The overall goal of this procedure is to quantitatively determine the influenza viral hemagglutinin and neuraminidase content in vaccines using antibodies targeting conserved epitopes. This is accomplished by first identifying conserved epitopes. The second step of the procedure is to dilute the reference and vaccine samples to improve the detection sensitivity. The third step is to assemble the slot blot apparatus and load the samples into the slots. The fourth step of the procedure is to detect the HA or NA on the membrane using their respective universal antibodies. The final step of the procedure is to quantify the amounts of HA or NA by densitometry. Ultimately, results can be obtained that show HA or NA quantified against their respective reference antigens.
Hi, my name is Anwar Hashem. I'm a PhD student in Dr. Sean Lee's lab.
Hi, my name is Caroline Gravel, and I'm a technician in Dr. Sean Lee's lab at the Center for Vaccine Evaluation at Health Canada. Today, we will be demonstrating a slot blot procedure used to quantify the HA and NA antigens of influenza viruses. The main advantage of this technique over existing methods like single radial immunodiffusion, SRID, or physicochemical methods is that it does not require strain or subtype-specific antibodies, contrary to SRID, or sophisticated instruments, like physicochemical methods. In addition, because these antibodies target the only universally conserved epitopes of HA or NA, they are useful as basic research tools for interested investigators. Generally, individuals new to this method will struggle because they may encounter technical issues, such as difficulty to blot precipitated vaccine samples, leakage of samples between slots, and loss of vacuum during the blotting step, as well as high background in diffuse